foreign students are in the lecture today? Hello? If any of the foreign students are in the lecture, please respond.
Yes, ma'am.
Okay, some of the foreign students are in our lecture. So today I will conduct my lecture in English. Can you hear me?
Yes, ma'am.
Okay, good. Then I can start with a small or quick recap of our previous lecture. Last class we talked about the isochromosome and diplochromosome. Isochromosome means the chromosome which have the similar arm containing the same genetic constitution. Okay, they are originated by the misdivision of the centromere. And diplochromosome is the chromosome which has double chromosome strand because of duplication of the chromosome without any cell division or division of the centromere. Today we will talk, we also talk about the B chromosome. These are the extra chromosome. Shilpi Rani. Shilpi Rani. Shilpi Rani, could you please? Yeah, thank you. B chromosome is the extra chromosome outer than the karyotype. It's called B chromosome. Today we will talk about it's called Lamb-Rush chromosome. Okay, Lamb-Rush chromosome, you can Shubhan, ID number 18021. Okay, thank you. Lamb-Rush chromosome. Lamb-Rush chromosome, from the word we can make, or we can guess actually how it looks like. It looks like a brush. You can see the brush to wash or for cleaning the lamb. Yeah. So the chromosome which looks like the brush of the cleaning the lamb is called Lamb-Rush chromosome. Okay. Now, see, if we see the definition, Lamb-Rush chromosome means the chromosome looks like the brush of lamb. Okay. It was first discovered by the Fleming in 1882, and the name was given by Ruckert in 1892. Okay. So the chromosomes which looks like a brush. It can be looks like a bottle brush, you know how the bottle brush looks like, or the brush of cleaning the lamp is called the Lamb-Rush chromosome. Of course, this is also a special types of chromosome because we know the chromosome actually have two strands with the genetic constituent, but if the chromosomes looks like the different than the karyotype, it's called the special types of chromosome. This chromosome actually huge. It's a big, very big, large chromosome. That's why sometimes it also called giant chromosome. Okay, it a very big size of chromosome than the normal compared to the normal chromosome. That's why it's called giant chromosome. So what actually exists in the Lamb-Rush chromosome? You see, this Lamb-Rush chromosome has the telomeric loop. It looks like, if we compare with it is a bottle brush. If you compare, it's called telomeric loop. This is the telomeric loop. Telomere means the end of a chromosome. Yeah, the end part of a chromosome is called the telomere. So in the telomere, there are some loops. It's called telomeric loop. And side loops in the arm of the chromosome, there are also have some loops, or not some, many loops. It's called side loop. And a chromatid without loops, there are also have some chromatid without any loops. Like you see, there has no loops. Could you please, all the students, be careful about the microphone? I am really very annoyed with the microphone. Tomal Shorkar, please mute your microphone. There has the Okay, can you hear me?
Yes, ma'am.
Okay, good. So there are also without any loops. There are some parts without any loop. It's called the chromatin without loops. So this is something interesting, yeah. Loops. First time I think you heard about the loops in the chromosome. I think did you heard about any loops in the chromosome? Maybe not. Okay.
No,
Let's. Okay. Loops. This is the special types of characteristic of these types of chromosome. These types of chromosome have many loops. Now, why they makes loops and what's the function of the loops? We will discuss about it later. Okay, let's see occurrence. That depends where we can find these types of giant and lamb-brush types of chromosome. It found in the growing oocytes of the most animals except the mammals. Mammals, you know the mammals. We are the group of mammals. Huh? So you can find in the oocyte. You know what is oocyte? Oocyte is the immature growing egg. It's called oocyte. So many animals you can find it like that lizard, frog, there's some earthworms. Lamb-brush chromosome of vertebrates, amphibians, reptiles, birds, and insects. This is the amphibians. Okay. Now, amphibians, these are the amphibians. The frog. Frogs are the group of the amphibians. And these are the reptiles, crocodile, lizard, cockroach. They are all under the group of reptiles. We also can find in the birds, insects, the growing oocytes. Oocyte means developing egg. Mostly in the egg yolk, these types of chromosomes are found. Chromosomes transmit into the lamb-brush form during the diploid stage of the meiotic prophase 1 due to an active transcription of many genes. Certain chromosomes stretch out of the large loops of DNA causes the chromosome to resemble a lamb-brush shape. So now we will know how this chromosome converted to the lamb-brush shape. Actually, during the cell division, you know what happens during the diploid stage of prophase 1? Diploid stage chromosomes started to pair, you know, they started to pair. During the S phase, that means synthesis phase, the chromosome duplicated, yeah, and when they started to cell division, during the diploid stage, some of the segment of the DNA, they make the loop shape. That means if you see this is the chromosome, you know, normally the chromosome coiled around the histone, we know that, yeah. Did you read or get the lecture about the structure of chromosome? Yeah, there are some histone protein. Normally the chromosomes are coiled in that stage, like in that way. This is the okay, I should forget to start the recording of the shit. I forget to record. So okay, so I'm sorry that I forget to start the recording of our lecture today. So now I started the recording. Okay, what we were talking about, normally the chromosomes are coiled around the histone. Okay, this is the, assume that this is histone and this is the DNA segment, DNA fiber. When the chromosomes are in that stage, around the histone protein, they are inactive. They cannot transmit or they cannot work. For acting or when they want to transmit or started the transcription procedure, they uncoiled around the histone. In the in the diploid stage, some of the segment of the DNA needs to be transcribed to produce the mRNA. For that reason, they uncoiled, and they, for work transcription process, they make the loops. Okay, they make these types of loops. That's why so many loops. They produce many loops. In that way, during the diploid stage, the chromosome get many loops because of the extension or stretching of some DNA segment, and causing chromosomes to resemble as a lamp brush shape. Okay. Found in those cells to which produce a lot of RNA, and the cytoplasm and the nuclear volume increase. The some cells which needs a lot of RNA, and for this reason they produce many loops and they get this structure of lambda shape. And in that way, the chromosome size centromere in the cytoplasmic nucleus become large, and the cell also become large. Okay. Characteristics. I had told you.
Can you repeat the?
I didn't get your answer.
Can you repeat the point again?
Okay. The chromosomes. There are some cells. There are some cells which need to produce a lot of mRNA. For this reason, during the diploid stage of Prophase 1, they extend, that means they uncoil and extend and produce the loops to produce the mRNA. If they are coiled around the histone protein, they cannot produce the mRNA. So for producing mRNA, they unpaired or uncoiled and produce the loops to continuously producing the mRNA. So some cells needs a lot of mRNA. That's why they produce the loops and the lambda structure to produce a lots of mRNA. And due to this reason, for producing this lambda shapes and the mRNA, the cell structure or cell size or nucleus become big. Yeah, because the lambda chromosome is a giant chromosome. If there is something giant inside the nucleus and produce a huge amount of mRNA, then the structure of the nucleus increase compared to the normal structure. Okay.
Okay, ma'am. Thank you.
So characteristic, as I told you, this is large. Okay, and larger than the salivary gland chromosome. We will talk about the salivary gland chromosome in the next topics. And salivary gland chromosome is also a giant chromosome. It also special types of chromosome. So it's all bigger than the salivary gland chromosome. Main axis has a row of granules known as the chromomere. You can see the structure of a giant chromosome. It is a segment of pair of two chromatid. Okay. Pair of two chromatid. Pair of two chromatid. They paired during the zygotin or diplotin. What is called? Leptotin, zygotin, diplotin, pectitin, diplotin diakinesis. During this stage, we know that the two non-sister chromatid exchange their segment by crossing over. This is the crossing over side, this is the crossing over side, this is the crossing over side, and this is the crossing over side. And these are two chromatid of a lamb brush chromosome. You see they looks like the brush. They have many loops, and the loops are created by producing many chromomere. Chromomere looks like the granules. Looks, these are the granules, and these granules are stand side by side and attached with the chromatin. Okay, these granules are called chromomere. This is the axis. It is a chromatin fiber, okay? And they are in the chromatin fiber. In some places, the chromatin fiber are dense and produce a granule-like structure. And this structure is called chromomere. And from chromomere, some segments are extended and produce the loops. Yeah, these are the loops. And every loops contain the repetition of same genes. And they produce the same structure in both sides. It's called the mirroring image of the loops. The genes which are present here also present here, and the genes which are repeated, many, because they need to produce many mRNA. Contains the main axis whose chromatin fiber DNA gives out lateral loops. These are the chromatin fiber who gives out the lateral loops. Throughout the length, there are many lateral loops. And you see each loops has two sides. This is one side and this is other side. They are the mirror image. They contain the same genes and also the same structure. And they are attached by the granules side by side. And they produce the Lambda structure ultimately. Okay. The chromosome process elasticity. That means they can stay, they can change their structure and they can stay for a long time. And after that, they also can change their structure. At the beginning of meiosis, when DNA replication is completed, the homologous pair lie immediately next to each other and form characteristic structure composed of 4 loops. That means after the replication procedure, before the meiosis start, the replication started and replication finished. After the replication, the chromatids, they produce 4 chromatids, and the 4 chromatids, they stand side by side and they started to make this structure, loop, chromomere, and ultimately the structure of a Lambda. Okay. Okay, this is the big picture of the Lambda chromosome. As I told you, these are the loops, these are the chromomere, these are the DNA fiber. The DNA fibers are dense and produce the chromomere, and in the chromomere, 2 loops are come out by extending of a segment of DNA. Yeah, and in this structure of loop, only the loops which only the DNA or genes which are located in the loops, they produce the mRNA. But the DNA which are in chromomere or not in the loops, they cannot produce any mRNA, or mostly they do not involved in the transcription process. Characteristics of loops. The number of loops varied. It depends on the chromatin material. If the chromosome have huge number of chromatin materials, they can produce many loops. If they have small number of chromatin materials, they can produce small number of loops. It depends on the how much genetic information on genetic material or chromatin material exists in the chromosome. Okay. Loops are too small and fragile in nature. They are very small, but you can see by the microscope, very high power microscope, you can see. And they are fragile. They cannot persist for a long time. They produce and they synthesis and then they again disappear. Okay. The number and position of loops remain constant in the same chromosome, but they are fragile, but the number and in which location they will produce the loop, this is consistent. Yeah, in a special types of chromosome, in one chromosome. For example, if chromosome number 1, it has 100 genes, maybe they can produce 100 loops. Okay. In chromosome number 2, for example, they have 1,000 genes, then they can produce 1,000 loops. So this is consistent in the chromosome according to the number of chromatin material. A given loops always contains the same DNA sequence and it remains extended in the same manner. a given loop. A loop has the, as I told you, the loops have same types of genetic DNA sequence, and it repeats, same DNA repeats, and extended in the same manner. The two sides, it called the mirror image. The loops of the paired chromosome form mirror image structure, as I showed you before. Transcription occur either along the whole loop or at a part of the loop. The transcription can be occurred in the whole loop or in a part of the loop. This is the loop. This is one loop, and you can see that red color at the transcriptive mRNA. Okay, the loops are involved in the transcription process to produce red color mRNA, and you can see the arrow, the direction of the transcription procedure. So transcription can be occurred in the whole loops or in a part of the loops. Okay. These types of loops have very high rate of RNA synthesis. You can see so many RNA are producing, and the production of RNA synthesis is very high rate. The rate of RNA synthesis because of the repetitive of the gene sequence that I told you. That's in the loops, the genetic sequence are repeated in several times. That's why the RNA synthesis rate is very high. Most of the genes present in the DNA loops are being actively expressed. That means the genes which are located in the loops, they are mostly genetically active. The majority of the DNA remains highly condensed on the axis, where the genes are generally not expressed. The DNA which are not in the loops, which are exist in the DNA axis, that means main axis where chromomere and the DNA fiber are exist, they are not expressed. A typical loop might contain between 50,000 to 200,000 nucleotide pair of DNA, although loops of millions of nucleotide pair also been suggested. That means in the DNA segment, that means in the loops, there may be 50,000 to millions of DNA. Okay. Chemical composition. What chemicals are actually exist in these types of chromosome? That I told you, the Lamb-Brush chromosome have two things or two components. One is called main axis where the chromomere, that means granules and DNA fibers are attached with each other. It's called the main axis. And the loops where the DNA are extended out and produce the DNA loops. Okay. These are two main things or main structure of the Lamb-Brush chromosome. So what chemical components are exist in these two types of structure? So main axis is produced by the DNA chromomere. So of course it has the DNA, and also it can have the protein to produce the structure. That means histone protein. Yeah. Loop, what can be had in the loop? Of course, it has the DNA because the loop is produced by the extension of the fiber of DNA. So it must have DNA. And the loops are active for the transcription procedure. So they must have the RNA. But main axis do not have the RNA because I told you that main axis mostly do not involved in the transcription process. They are not active. Only the active DNA are exist in the loops. So they are active. They involved in the transcription procedure. So they must have the RNA. And also, as they involved in the transcription procedure, so from mRNA to proteins are produced, so they also should have the protein. Okay, so Lamb-Brush chromosome has two main structure, main axis, loops. Main axis has DNA, protein, and loops have DNA, RNA, and protein. Okay, these are the two main chemical components of the Lamb-Brush chromosome. So function of the Lamb-Brush chromosome. Of course, they are chromosome, they have DNA, they must involved in the protein synthesis. This is the normal thing, everybody know the functions of chromosome. Okay, protein must be synthesized in the Lamb-Brush chromosome. For synthesizing protein, of course, they have to first go through a transcription procedure. By the transcription, they produce RNA synthesis or RNA. They also produce the nuclear material, increase the nuclear material. I told you that in the cells which have the Lamb-Brush chromosome, they have the high rate of RNA synthesis because the Lamb-Brush chromosome loops have repeated of the genes. Due to the repetition of the genes, the rate of the DNA synthesis is high. That's why the nuclear material increase. Produce nucleolus, which are essential for the formation of embryo. I told you at the beginning that where actually mostly the Lamb-Brush chromosomes are found. Mostly the Lamb-Brush chromosomes are found in the oocytes. Oocytes means in the growing eggs or in the growing cells which started to be produced the embryo. So for formation of embryo, this egg yolk, that means ডিমের কুসুম আমরা যেটা বলি, egg yolk has the Lamb-Brush chromosome, and these are very important for producing the egg yolk of the egg, a fertilized egg. They are essential for the producing the egg material as well as the embryo development. Okay. This is all about the Lamb-Brush chromosome. So we get to know different types of special chromosome. This is also called the giant chromosome. It has special characteristics, main axis, loops, and they have high rate of mRNA synthesis, and mostly they are found in the developing oocytes or egg cell, especially in the egg yolk. Okay. Now we will talk about another types of giant chromosome. It's called polytene chromosome, or also called salivary gland chromosome. That I told you in the polytene chromosome that Lamb-Brush chromosome is larger than the salivary gland chromosome. But what is salivary gland chromosome? The other name of salivary gland chromosome is called polytene chromosome. And why it is called salivary gland chromosome? Because it is found in the salivary gland of Drosophila melanogaster. You know what is Drosophila melanogaster? Drosophila melanogaster is the scientific name of fruit fly. I think everybody of you know what is the fruit fly. And summer is coming, you know, you will get or you will find a lots of fruit fly in the fruit, in the mango, in the jackfruit. Yeah. So these types of insects, that is fruit fly, the salivary gland contains some giant chromosome, and these giant chromosomes are called salivary gland chromosome or polytene chromosome. Why? The name of this polytene chromosome, I told you that whenever you get any word, a scientific word, whatever the word, always try to understand what it is by the name of this word. Okay. Poly means many and teen means strand. That means the chromosome which have many strands is called polytene chromosome. So how a chromosome can get the many strand? How? One possibility is that the chromosome do not contribute in the cell division, or the centromere do not break down when some chromosomes are replicated during the synthesis stage. Okay, we know that during synthesis stage, a stage of cell cycle, all the chromosomes contribute in the replication procedure. In the replication procedure, single chromatid chromosome become two chromatid. And during the cell division, metaphase stage, the bichromatic chromosome become single chromatid again by the breakdown of centromere. Everybody know this stage. Again, this single chromatid become bichromatid at synthesis. Again, it break down in the metaphase stage. Then it convert bichromatid to single chromatid. This is the normal procedure of cell division or maintaining the chromosome structure. But if a chromosome continuously replicated at S phase, that means synthesis phase, but do not break down the centromere in the metaphase stage, that means the chromosome strand number increases. If it repeated, then it will get many chromatid, but it have only one centromere. Okay, these types of chromosome are called polygene chromosome. I think you got what I want to say, that what is actually polygene chromosome. The chromosome which replicated during the synthesis phase, but do not involved in the centromere breakdown at metaphase stage of cell division. Then it has, when this procedure is repeated, then a chromosome get a lots of many number of chromatid, but only one single centromere. This type of chromosome is called polygene chromosome. And is found in the salivary gland of the Drosophila melanogaster, that means fruit fly. That's why it is also called salivary gland chromosome. And also sometimes it's called giant chromosome because it's big structure or big chromosome. Okay. First reported in Balbiani by the Balbiani in 1881, and the name was given by the Kolar due to the occurrence of many chromosomes or many chromatid. Origin: specialized cells undergoes repeated round of DNA replication without cell division. It's also called endomitosis. To increase the cell volume, forming a giant polygene chromosome. Polygene chromosome form when multiple round, 9 to 10 repetition of the sister chromatid, that means synapted together. That means they are together. It's called synapted. That means 9 to 10 round of synthesis, but the cell did not involved in the cell centromeric division in the metaphase phase. It is also called endomitosis. That means mitosis occurred, but the centromere didn't break down in the metaphase phase. It is called endomitosis. Okay. Where we can find these types of actually chromosome, special types of chromosome or giant chromosome. I told you that it first found in the salivary gland of the salivary gland of the fruit fly. That's why first it was named as salivary gland chromosome, but also it is found in many other organisms. For example, many insects are of the order Diptera. What is Diptera? I think you already get to know in the entomology that the Diptera means dip, di-tera. Di means two, tera means wing. When an insect had two wings, this is called Diptera. Two wings have in the fly. You know, this is fly. They have two wings. This types of insect group is called Diptera. The salivary gland, gut, trachea, fat body cell, Malpighiani tubules, epithelium, rectums. These types of organs have this salivary or polygene or giant chromosome. We also found in the angiosperm. What is angiosperm? Angiosperms are the plants, group of plants, where the seed is covered by a shell or by the fruit or by the some structure. Covered seed is called angiosperm. The other word is gymnosperm, which are where the seeds are open. Seeds are not covered by any types of structure. For example, pine, palm tree. In these types of plants, the seeds are open. They do not covered by any types of structure. This types of plant group is called the gymnosperm. Okay. In the angiosperm also I have some salivary gland chromosome or this polytene chromosome. Protozoa. Protozoa is the cells. It's called. You study about it in the HSC biology. Protozoa is the first found or cells without any structure. Okay. They have the lower animal. It's called. It is things that the animal kingdom started from the protozoa. These types of this is the protozoa. They do not have special structure and not well organized nucleus. They have the giant chromosome. Okay. Carcinomas. Looks. Sometimes we found some outgrowth in our body, infection. Sometimes we call maybe there has the infection. Okay. Carcinomas, actually the term comes from the cancer. Sometimes when some skin has some cancer disease, they produce some bubbles, and from the bubbles some infection in the body, and these types of infection there are some cells where these types of polytene chromosomes are exist. Sometimes it can produce the cancer. That's why it's called carcinomas. It's a disease. Okay. In these types of disease, cell sometimes have the polytene chromosome. Okay. So we will talk more about the polytene chromosome. We have to know the occurrence, we have to know the structure, we have to know the function of the polytene chromosome. So I think for today, it is all about my discussion about the Lamb-Rush chromosome and part of the polytene chromosome. Okay, now we have 20 minutes to discuss about the questions. If anybody of you have the question, please let me know. Any question?
ম্যাডাম, সেলে যখন এমআরএনএ প্রচুর পরিমাণে প্রয়োজন থাকে তখন ম্যাডাম ক্রোমোজোম গুলো ডিকয়েলিং করে ল্যাম্ব-রাশ স্ট্রাকচার তৈরি করে, এরকম তো?
যখন একটা ক্রোমোজোমের অর্থাৎ যে কিছু কিছু সেল, যেমন ধরো এগ ইয়ক, some egg yolk during development procedure, they need to produce huge amount of mRNA. Not all chromosome produce the Lamb-Rush structure, not all. Some animals have this types of special types of chromosome. During the diploid stage, they need to produce the huge amount of RNA. Then they get the structure of Lamb-Rush chromosome, and after the cell division, they go to the normal structure. Okay, but it doesn't mean that we also have the Lamb-Rush chromosome. No, we do not have the Lamb-Rush chromosome. But in some animal, for example, frog, crocodile, reptiles, some birds, they are egg yolk, they have some special types of chromosome called Lamb-Rush chromosome. During diploid stage, they need to produce many mRNA for transcription procedure. Then they get this loop structure, ultimately the Lamb-Rush structure. Okay.
ম্যাডাম এমআরএনএ যেগুলো প্রয়োজন সেগুলো তৈরি করা হয়ে গেলে পরবর্তীতে কি আবার নরমাল স্ট্রাকচার তৈরি করে?
Yes, yes.
কয়েলিং করে?
After his activation, after his activity, when he do not need to produce any types of mRNA, then he go back to the normal structure. But this types of only this special chromosome has the capability to produce the Lamb-Rush structure. Okay. That's why it is called they are elastic. They are highly elastic. They have high elasticity, and also called the loops are fragile. They can break down and very unstable. Yeah, they synthesis and then go back to their normal structure. Okay. Any more questions? So it looks like that you all enjoyed the class and understand everything. Yeah. ID number Afsana 1902400. Afsana, are you there? Good.
Yes, ma'am.
Okay. Then Fahmida, are you there?
Yes, ma'am.
Good.
ম্যাম ল্যামডাস
Really?
ফরমেশনটা কি স্টেজে হয়?
Sorry?
ম্যাম ল্যামডাস ফরমেশনটা কি ডিপ্লোটিন স্টেজে হয়?
Huh. During the diplotin stage.
ঠিক আছে।
I can see all are female. Jahangir Rajon, are you there?
Yes, ma'am.
Good. I'm looking for some criminals who are activated in the online but not hearing me. Let's see if today I can get some criminals here. Shoma. Yes. Shoma. Okay. And how about the foreign student in my class? Where is the foreign students?
Yes, ma'am, we are listening.
Okay, where are you from?
I am from Nepal.
Okay, can you get my lecture? What I'm talking about, and do you have any problem to understand my lecture?
No, ma'am, I understand clearly.
Okay, good. And I think the Bangladeshi students also do not have any problem to get my lecture. Okay. Are you okay that I'm the lecture in English? Okay, then it's fine. I will take only one more lecture, then my lecture will be completed for the level 2 semester 2. Okay, so I think no more questions. Yeah. So if there is no more question, then we can finish our lecture for today. I hope you all will be fine. And if further have any question, you can send me an email to my email account. I will reply